This is episode 114 of the Immunology Podcast, The Importance of Investing in Science with Dr. Eric Vivier. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoyed the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Eric Vivier from Aix-Marseille University on the podcast to talk about the importance of investing in science. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Do you work with human immune cells? Learn how to isolate highly pure immune cells and efficiently process human samples with a free, on-demand course on human immune cell isolation. To learn more and register, visit stemcell.com/humanimmunecourse. Hey, Jason.
Hello.
Grüß Gott, as the Austrians would say.
I'm back in the States.
I'm back in Amsterdam.
Yeah. It took me like 4 days to fix my time zone. It wasn't too bad.
It's pretty hard, huh? The time difference, but I'm glad to see that you're back.
Yeah, I was like waking up and then getting sleepy at weird times. It's okay.
Yeah, that's how it goes. It was fun, right?
It was. Oh, I loved it. I was in food heaven.
Like, I love that schnitzel.
And the sausage and the beer and the cappuccinos and the chocolate.
On the cake.
Yeah, the pastries. So good.
Yeah.
So good. All right. So, we're talking science funding today. So if I could give you unlimited money, what would you go do with it, Brenda? For science.
For science.
Yeah. You can only spend it on a science project to answer one big thing.
Well, I have to say, and you're going to love this, how can we engineer a microbiome to be more healthy?
There you go. Oh, yeah. That's awesome. See, I'm not going immunology or biology. I would figure out how we could live on the moon. Because if you can solve all that, you solve a ton.
Fair enough. Why not Mars?
Closer.
It's too far. Too complicated.
Yeah.
All right. Living on the moon would be fun.
Because you got to solve space elevators. You got to solve environmental engineering. There's all types of things you got to solve to do that.
Well, they did not have unlimited funds, I assume. But, a lot of people have been doing stuff and publishing papers. So why don't we talk about that? And what you got for me this week?
Oh, I got a couple of good ones for you. The first one came from some guests, a listener recommendation, actually. In Cancer Cell, it is called Genome-Wide CRISPR Screens Identify Critical Targets to Enhance CAR NK Cell Anti-Tumor Potency. Co-first authors are Alexander Bierderstadt and Rafet Basser, and the corresponding author is Katayun Rezvani. And it came out August 21st, so just a little bit ago. So, high level, they were able to figure out how to do CRISPR screening in NK cells, which have been notoriously difficult to be able to get good NK cells screening into. And they say they do this from core blood-derived NK cells, and they developed a retroviral vector system, including a guide RNA expression cassette, and then electroporated in the Cas9 protein and selected with puromycin. It's kind of their approach. They use universal antigen expressing feeder cells, UAPCs, plus IL-2 as well in this thing. And they came up with a name for it, which I'm going to share just because it's a lot of acronym. It's called PRECISE with a capital P, lowercase r-e, capital C, lower I, capital S and E. And that's supposed to stand for Pooled Retroviral Library Delivery and Cas9 Electroporation in Primary Human NK Cells to enable systemic exploration of genetic perturbations. And while I think the paper is very cool, just because you can make an acronym doesn't mean you necessarily should in this case. It's very forced. It uses a lot of words in between each letter of the acronym, and it's a little harsh. But I like the paper, but yeah.
Well, that's your feedback to the team.
Reading it. Anyway, so they're able to do this. And they do a whole bunch of stuff with it, right? And so, like, you know, if you're an NK person, you should read this paper for sure, not just an NK CAR T person. But they looked at, you know, so they used a library, they did a transcription factor library with 1632 transcription factors and 11,364 guide sequences, and found transcription factors that when you knock down either made the cells less viable or more viable or have more or less anti-tumor proliferation. And they did an unperturbed screen with high coverage delivery of 77,000 plus genome-wide library targeting over 19,000 genes. So they did that too. So it's just this huge multi-omic process with a whole bunch of stuff shoved in and next-gen sequencing. It's very high throughput and very fun, very impressive in what they did. So they did a whole genome, and then they validated screening hits using publicly available data sets. So they saw something interesting. They went and looked at human data sets to see if they had similar signatures of either efficacy or something. So for instance, they saw that, they saw that the gene signature in the top 5 hits in re-challenge screens were significantly upregulated in tumor infiltrating and circulating NK cells in pancreatic patients, cancer patients, versus healthy donors. So they're able to recapitulate this in the real world. They mapped regulatory nodes for NK cell programming by looking at string and Go databases and mapping on top. They look at genes that overcome the tumor microenvironment based immunosuppression. They put this on top of various models to detect the effect of these things, modulating resistance to TGF beta, overcoming acidity-induced immunosuppression, making them resistant to hypoxia. But then they looked at some fitness for clinical manufacturing, and I thought this was really cool. They looked at which genes affected manufacturing fitness. They came up with a list there. And then they were able to look and compare this to known things in other cell populations, CRISPR screens in primary human T cell and CAR T cells, and saw some of the same things, positive regulators like IRF4 and IL2RG, negative regulators like TNF-AIP3 and FAS. So they kept seeing patterns. And then they prioritized some of these MED12, ARIH2, and CCNC were top candidates for improving NK function. And they did some functional studies by creating knockouts in CAR NK cells and having better function. So they took it all the way down to like, I made a better NK cell. But it's just an insane amount of gene editing with entire genome mapping of knockouts. And they did it in everything from cell systems to eventually cancer models in mice. And then back whenever they could, they went back to a human data set and tried to compare it.
Wow. That sounds like a lot of work.
Yeah, it was a huge amount. Do you got anything with CRISPR or CAR cells or anything?
No.
No segue for me?
No segue.
Oh.
Yeah, I know. I'm sorry. I do have one fun story about synthetic biology. So I guess it's not gene editing, but it is kind of genetic engineering in a way. But I'm going to start with a different story. And I'm not sure if this is cheating a bit, because I did comment a bit on this story during our coverage of IUIS. But the paper was published like a week after IUIS. And I kind of want to talk about it again because I really think it's pretty cool. So we'll see. So this, my first story of the day is a paper published in Nature, 27th of August. First author, Nadine Serran, from corresponding author Nicolas Gaudencio They are at the Toulouse Institute for Infectious and Inflammatory Diseases in Toulouse, France. And the paper is called Maternal Stress Triggers Early Life Eczema Through Fetal Mast Cell Programming. And as I mentioned, I already kind of touched upon this presentation during IUIS, but this is nice because, they got their actual publication out, and it gives me a chance to revisit this very interesting idea, which is that prenatal stress of pregnant mice in this case, can result on kind of longer term immunological sequelae to the offspring. And this is particular around a phenomena known as pediatric eczema, which it's found in children that have this extra kind of sensitivity in their skin, and they get rashes from diaper rashes, I guess, or from friction, or they get, and this is something that apparently it kind of improves with age. So it's temporary, maybe for the first years, months, but there's no clear like where is this coming from. It's just, and whether there's a link with prenatal stress. And so in this study, what they do is they have a mouse model of pediatric eczema, and they show that there's actually a correlation, kind of a mechanistic correlation between stress and the development of this disease. So basically, how the model works is that they have, pregnant female mice, and they stress them out by shining very bright light on them at the end stages of their pregnancy. And apparently this kind of increases their cortisol levels and this stresses them out. And they find that the offspring of these poor stressed dams, as the female mice, have this dysregulation in the skin, in mast cells in the skin, and also certain increased sensitivity to pain. There's this mechanosensor assays in which they basically poke mice and to see how much they feel mechanical poking. And they show that basically the offspring of stressed mice, they have a higher tendency to develop eczema when they kind of increase the friction, and they kind of irritate the skin, and they have this regulation in the pain perception. And so they have this model that works, and they can in a way imitate this phenomena that they kind of this human phenomena, and they try to understand where is this coming from. Why are the stressed mice having offspring with this differences? Basically, they show that there seems to be a separate. One thing is the increased sensitivity, and the other thing is the development of the eczema. So if we focus on the eczema part, it is the overactivation of mast cells in the skin. And what they study the source or which kind of mast cells there are, and they show that this only happens in the first few weeks of the life of the mice, and then it goes away, suggesting that there's something about developmental stages that affects this this kind of long-term effect from in utero. And they show that there's these mast cells that have a yolk sac origin that then they get kind of replaced by hematopoietic regular hematopoietic origin mast cells. But it's these mast cells that they get in a way primed, or they become more activated in utero as a consequence of the increase of cortisol in the amniotic fluid of the stressed pregnant mouse. Basically, that's kind of the mechanism that they see. And that this really, they do studies in which they look at they study different cells, and they really pinpoint that this is the connection between the stress and the later eczema that the mice develop. And so, basically they show that after a few weeks, I think by week 24 of these mice, this pathogenic phenotype reverts naturally, and this coincides with kind of the loss of these fetal mast cells um, and replacement by ones that are they're not derived from the yolk sac, so they're not of fetal origin. So I thought it was pretty cool because, yeah, it's a very direct connection between, yeah, something that happens to the mother and in a very specific way that affects the child. And the idea is that this could also explain some of the effects that you see in human newborns. And they do have a bit of, they pinpoint certain receptors that are sensing the cortisol, and they show that similar to what they see in these mice, these receptors are also present on fetal mast cells in babies. So probably they could also respond to cortisol in a similar way. And it seems that atopic women have more tendency of having children with this eczema, and that this atopic pregnant women have higher circulating cortisol, and that probably might be a mechanism by which these babies develop this increased sensitivity or this increased chance of getting eczema, but only pediatric one, right?
Yeah, it's interesting that it's just the cortisol receptor again. I think like everything gets blamed on cortisol.
Yeah. It it really makes me think, you really need to lower those cortisol levels because there's so many things nowadays. They're connecting cortisol to so many immune cells. It's pretty interesting.
Yeah, I think it's surprising that there's such a clear effect. But I like the fetal angle to it, with them going away to help explain the cut.
Yeah. That's also very interesting.
Well, you want to know what else is stressful is unfolded proteins. And we're going to talk about the unfolded protein response.
All right.
In immunity, it is unfolded protein response transcription factor. XBP1 suppresses necroptosis-induced colitis by reinforcing the gut mucosal barrier. Our first author is Goksu Goperkaya. And then last author is Manolis Pasparakis. So my guess is this isn't in Nature because some of this is already known. And so we know that XBP1, Xbox binding protein 1, deficiency can induce colitis spontaneously, although it's not particularly strong. We know caspase 8 intestinal and FAD intestinal knockout mice have increased, they develop pretty bad spontaneous colitis. So we know that as well due to necroptosis, essentially. So they have heightened necroptosis, but interestingly, the XBP1 doesn't have ileitis where the other two do. So they cross all these mice together in a tour de force of mouse breeding and learn that, XBP1 on top of either caspase 8 or FAD knockout have way worse colitis. So that's the first thing. It exacerbates it. But if you add on RIPK3, which is downstream to induce apoptosis or necroptosis, that ameliorates both of these differences. So it reverses it. So that's a triple knockout now we have. A tissue-specific triple knockout. So you got 4 different alleles going on here. So, I do not know how many mice they bred, but they bred a lot of mice here. They look at the genes. In the XBP1, they notice doesn't change the ileitis, which is important. They inhibit apoptosis and necroptosis, and that doesn't do anything for the XBP1's loss of Paneth cells. They see there's this loss of Paneth cells in XBP1 knockouts, and that adding in either a RIP3K or an MLKL knockout doesn't reverse it. So then they look at TNFR1 signaling because IESP specific TNFR1, so tumor necrosis factor receptor 1 deficiency, mitigates colitis development in the caspase ACE model. And that is important in the worst colitis in XBP1. So when they do that triple knockout, they get better. So they're finding that but only in the large intestine. So again, the ileum is spared this effect. And they try to figure out why. So they do some RNA-seq, and there was less MUC2, which is the main mucus protein. And so there's less MUC2 gene. And then they do all the staining and all the models and show, yeah, indeed, there is less mucus being formed. And they see that there's more bacterial translocation in the knockouts. So the mucus layer being worse is leading to more bacterial translocation, which exacerbates disease. And if they inhibit necroptosis, prevented the bacterial translocation and inflammation with, again, a RIP3K or RIPK3 knockout. Then they figured out it's goblet cell intrinsic that's driving this. So they did this by doing organoids that they differentiated specifically towards goblet cells and saw this was an intrinsic process in the goblet cells producing less. And they looked at the signaling there, and they found that ablation of XBP1 in the IEC leads to activation of the inositol requiring enzyme 1 alpha, IRA1 alpha. That had increased phosphorylation, which is known previously, and that mediates unconventional splicing of unspliced XBP1 to spliced XBP1. And so IRA1 can then degrade a certain pool of mRNAs by its endonuclease domain. And so they thought, and this is called a process called RID. And so long and short, they thought that MUC2 could be targeted the RID process and activation of IRA1 in XBP deficient. So getting rid of XBP1 leads to more RID process, and that leads to degradation of MUC2.
Right.
And so they then link that all up. And then, just because there's never one gene, there's anterior gradient protein homolog 2, AGR2, AGR2, and its absence leads to reduced MUC production before MUC2 production, and XBP1 is thought to regulate this in lung. And lo and behold, in lung deficiency, they had impaired AGR2. So there's two different mechanisms by getting rid of XBP1 results in decreased MUC2. So it prevents activation of IRA1 beta and inhibits IRA1 beta mediated MUC2 mRNA decay by inducing AGR2. So XBP1 prevents activation of this RIG process and thus prevents degradation by inducing the AGR2 gene. And so they map this all out. And so the long and story short is that XBP1 is a big controller of the mucin protection layer. And then in the absence of that, the necroptosis goes extra wild because you have more bacterial translocation driving even more necroptosis. And then the intestine falls apart.
They also do caspase knockout?
Yeah, that's how you caspase 8 knockout leads to necroptosis.
Right.
Right, that's the baseline. So this is all done in caspase ACE or FAD. That's already known. They just did XBP1 knockout on top of it.
Because I'm looking at the abstract, the graphic abstract.
Right.
And the double knockout is even worse.
That's the whole point of the paper. They already the whole point is that it exacerbates it by getting rid of MUC2.
All right.
So XBP1 knockout on top of the no necroptosis system, the caspase ACE knockout or the FAD, leads to loss of MUC2 production, which leads to less mucus layer, which leads to more intestinal inflammation and worse disease.
All right. So I guess, yeah, having MUC2, it's important to keep that and XBP1 is involved in it.
Yep, XBP1 is important.
All right. So for my second paper of the day, it's a little bit old, I have to say. It's from almost a month old, but I really liked it, and I had bookmarked it before we went to Vienna, and I was like, oh, I really want to talk about this one, so I will. The paper is called "Expanding the Cytokine Receptor Alphabet Reprograms T cells into Diverse States." And first author is Yang Zhao from corresponding authors Anusha Kalbasi and Christopher Garcia at Stanford University School of Medicine. Yeah, both of them. And so Christopher Garcia's group is well known for doing synthetic cytokines and orthogonal cytokines. And so they've been kind of, I guess, just testing different combinations and seeing where they can get this technology. So basically, the idea of this synthetic cytokine receptors and their corresponding, orthogonal cytokines is cytokines are important for, the development of immune cells, for the signaling and for programming them into different things. And of course, one of the most, when it comes to cancer immunotherapy, probably one of the most spoke of cytokines is IL-2. And the receptor of IL-2, it has 3 subunits. The IL-2 alpha, also known as CD25, very well known, the IL-2 beta, and the IL-2 gamma, also known as the common gamma chain, which is a subunit that is actually shared across a bunch of receptors. So the gamma common chain can bind to different, a range of different co-receptors to make the receptors that respond to different cytokines. And this is kind of, well known that there's the signaling that most of the cytokines signal through what is known as the JAK-STAT pathway, in which you have JAK proteins that are associated to these different subunits of the receptors, and then they end up phosphorylating different combination of STAT transcription factors. And it is this combination that will, in the end, result in whatever gene activation this particular cytokine induces. So you have, for example, IL-2 is well known for inducing STAT5. The interferon alpha, interferon type 1 receptors will induce STAT1, STAT2, and then you have, and so on. You have different cytokines will generate the translocation of different STAT proteins to the nucleus and therefore initiate different genetic programs. So one of the things, so if in the context of, for example, cancer immunotherapy, if you want to, help out your T cells to, attack a tumor or survive, you might as well give them IL-2 because, IL-2 gets the T cells to proliferate, usually associated with improved performance of the T cells. But IL-2 has all these, if you have too much IL-2, can be quite toxic because it will activate T cells all over your body and give you a lot of undesirable side effects. So what they designed originally in the previous research is an orthogonal IL-2 receptor that basically only responds to an orthogonal IL-2. There are mutated cytokines and mutated receptors. Then if you have cells that are expressing this orthogonal IL-2 receptor, then you can, pump a lot of this orthogonal IL-2 and will only activate the cells that have this transgenic receptor. Therefore, it's kind of helping reduce the chances of target cell effects. So basically, you have this original version, in which is basically an IL-2 receptor that will recruit the same STAT proteins as the original IL-2 receptor. But then they thought, well, what if given that we can pair this gamma chain, will pair with many things, why don't we just start changing the JAK proteins of this receptor? This receptor is synthetic anyways. So can we get away with changing the intracellular domain, which will result in novel combinations of JAK and STAT receptor activation? And so basically that's what they do. And they show that they can just, mix and match these domains. And then by using the same orthogonal IL-2, they can induce progenetic programs that are associated with other cytokines. So they can induce, STAT1, STAT2, STAT6. These are all associated with other cytokines, but by using this orthogonal IL-2, this synthetic IL-2 cytokine. And basically what they look into is like, what happens? What does this help the cells? First, how does this change the cells? And then in the context of tumor immunotherapy, does this help? And so they show that indeed they can start associating, activating other STAT proteins different from IL-2, and that then they can really fine tune or change in a way the behavior of these T cells. And interestingly, they do both CD4 and CD8s. And in CD8s, particularly, is most interesting because CD8s are not going to express IL-4 receptor the way CD4s are going to. So they really can have some novel cytokine signaling in a cell that naturally would not respond to the cytokine. So in the case of CD8 cells, if they have, for example, a receptor and an orthogonal IL-22 receptor that generate by modifying the JAK signaling, they can actually, for example, increase the stemness and reduce exhaustion of the CD8 and of these engineered CD8 cells, which seems to enhance persistence and anti-tumor efficacy. And what I thought was pretty cool is that if they make an orthogonal receptor that has that will transduce similar to granulocyte, the GSCFR, which is a myeloid, is a receptor expressed on myeloid cells normally, but they can actually partly induce this program on T cells that it actually kind of gives T cells this kind of myeloid kind of expression. So they end up making things like CD14 and what is it? They have, it's like this myeloid-like hybrid that they generate. They upregulate phagocytic machinery and they can phagocyte bacteria. These are T cells, right? They maintain cytotoxicity, but they acquire some of the properties that are traditionally associated with myeloid cells because they are, upregulating, I think it's STAT3 and as a novel thing. And they also show that they can have IL-10 receptor, kind of this orthogonal IL-10 receptor on CD8 cells, and that this can also change the characteristics of these T cells. So basically, I think the bottom line is that first, it's really cool because you can push the T cells to do things that are not normally found on T cells. They can, for example, skew Th2. Basically, you can generate a new phenotype just using this orthogonal IL-2, which doesn't signal like IL-2. It makes what another cytokine would do. I don't know if I think it's clear what I'm trying to say. So I thought it's really cool because then it allows you to maybe try to fine tune the kind of cell that you want by adding the right intracellular signaling domains that you want. And you can always achieve this using the same kind of cytokine to stimulate these novel receptors. And this might, if you are smart enough about this, you might find the right combination of signals that a T cell you can make this T cell have that might make it a better, in this case, anti-tumor cell.
So everything is still a skid, Mark, and you can make one cell into another with the right cytokines. Yeah, I guess so. I guess that's how it goes. But it's kind of fun because then it really opens this. You can kind of. The thing is, skid marks it's nice to try to be able to really modulate the skid mark a bit. It's always going to be a skid mark, but it's nice to be able to kind of direct the skid mark towards what you're looking for. You want to be able to push it around.
Yeah.
That's interesting. All right. Well, with that terrible set of analogies that we really should stop, we're going to be speaking with Dr. Eric Vivier at Aix-Marseille University in just a moment. But before we get to that, Do you need your regulatory T cells for your experiments? With Immunocolt Mouse Treg Differentiation Supplement from STEMCELL Technologies, you can differentiate mouse naive CD4 positive T cells into regulatory T cells. Once isolated from mouse spleen, simply culture your CD4 positive T cells with a supplement and anti-mouse CD3 and anti-mouse CD28 antibodies. The Treg cells generated can then be used for your downstream experiments, including T cell suppression assays. Learn more about the mouse Treg differentiation supplement at www.stemcell.com/immunoculttreg.
So for our second part of our episode today, we are live from Vienna, Austria. Well, not live. Is this live? For this one?
We're recording on location.
We're recording on location. Yeah, that's true. It's not. So we are recording on location, and we are really happy to have physically in front of us for not very often this happens, Dr. Eric Vivier. Dr. Eric Vivier is a professor of immunology at the Center for Immunology in Marseille-Luminy and the Aix-Marseille University in Marseille, France. And we're really happy to have him around here because we're going to be talking today about the importance of funding science and of science reaching, students reaching basic science and how important it is to support this and for societies to fund science and understand the contributions that it brings to all of us. And so this is a collaboration that we have with IUIS, and we're really happy to be discussing this. Professor Vivier is also a member of the French Society of Immunology, and so it's part of the immunological kind of society environment in France. And it's really nice to bring these topics to the discussion. So Dr. Vivier, thank you so much for joining us.
Hello.
And here in Vienna.
Thanks for the invitation. It's a pleasure.
Thank you, Brenda, for not making me try to butcher French.
I know. I'm trying.
Much
That's okay.
appreciated.
I got your back. All right. So maybe I can start a little bit of background that is kind of, in a way, has inspired this conversation and the reason we wanted to have a minute to talk about this. So science lately has been on a kind of complicated time. There's been a lot of challenges, to put it mildly. And I think particularly everybody's very looking at what's going on in the US and the issue that science in general, science perception, and the kind of things that people can research, and more, in a more practical sense, science funding and what is the role of governments in providing the necessary resources for scientists to do their jobs. I think mostly one of the most kind of impressive numbers that we've seen in the last few months is the cuts that were proposed for the NIH. There was a proposed cut of $18 billion to the NIH budget. We was about 40% cut from around 47 billion total budget. And this was in the current administration a reflection of the rejection or the issues that we have now. And I think across the world, sometimes skepticism of science and in general, a less support from governments to scientists. And we are lucky that apparently is not going to happen. So there has been pushback from lawmakers in the US. And so I think for now, the NIH can rest a little bit more, be a little bit more sure that there's not going to be a 40% reduction in their budget. But I think this kind of events or this uncertainty around how governments fund scientific endeavors really makes us think about what is the role of science in society. And I wanted to hear, maybe we can start a little bit of a discussion around that. Also, in Europe, the situation is a bit different. So, of course, every country in Europe has their own science budget. I think ballpark figures show that the 40 billion that the NIH has is really large. And that's why everybody in the world was thinking about 40 billion is what is the whole science budget of Germany, which is one of the largest economies in Europe. It's larger than the 30 billion-ish that has the whole science funding in France, which is now the other big player in Europe. So it really shows that the numbers are in the NIH are pretty large. And although the European bloc is also trying to provide other sources of funding, we have the Horizon Europe budget that is apparently going to be increased from the 95 billion that was earmarked for the 6 years between 2021 and 2027 will be doubled ish around to 175 billion for the 2028-2034 period, which is for all of science in Europe, including, astrophysics. And so there is health, but there is also geology, ecology, material science. So everyone together is getting, so that really shows when you compare with 40 or 48 billion, which is what is currently going to be set up for next year's NIH budget. It really shows that still Europe has a way to go to be anywhere in the ballpark of biomedical research funding in the U.S. So I'm gonna shut up now. I'm gonna ask you, Eric, what do you think is the impact of these kind of volatility or the risk of having this problems, or this governments being able to defund science, or maybe in a more positive note, why should governments be earmarking this large amount of what feels like a lot of money for funding the research in their countries?
Yeah, well, funding of research has always been a problem worldwide, unfortunately. And I think it's important to remember that in societies which are based on knowledge, it's absolutely critical to fund research. And in particular, basic research. There are many examples showing that the application of basic research have changed mankind. I'm just going to take one example here because we are at the International Congress of Immunology, which is the one of monoclonal antibodies. Actually, it's timely, I think, also because in Nature, this week's Nature magazine, there is actually a very nice editorial on monoclonal antibodies, because we are celebrating 50 years of the discovery of these monoclonal antibodies by Cesar Milstein and George Koller, who actually got the Nobel Prize in 1974, if I remember correctly. And it's really interesting because they were not doing this kind of research for curing anybody, nor for any kind of application. What George Koller and Cesar Milstein wanted to do is to answer a very basic question on allelic exclusion in B cells, which is really niche, so to speak. And they came up with this technology of making monoclonal antibodies, and they published this Nature paper, not providing any kind of insight as for the future development of these monoclonal antibodies, but just to answer the question of allelic exclusion in B cells. And it's maybe interesting to remember also that at this period of time, the Office of Technology Transfer of Cambridge didn't think it was anything to patent. So there is no patent originally on monoclonal antibodies, believe it or not. I'm just going to give you now another, one number. Pembrolizumab, also called as Keytruda from Merck, is making, as far as I know, $15 billion dollars a year. Yeah. So I think it's interesting at different levels. First of all, again, it illustrates the fact that basic research can give rise to applications that we ignore at the time of the discovery. And that's obvious. You cannot predict of the application of something that you just discovered. And I think that another very good example is Louis Pasteur himself. Louis Pasteur, I would like to remind that to everybody, was very much known to be involved in industrial research, so to speak. He was very involved in trying to improve the manufacturing of wine, beer, and silk in France and in Europe before he contributed to the discovery of microbes and to the generation of vaccines. So I think that Louis Pasteur was really adequate to speak about the application of research. And he was saying there is no such a thing that applied research. The application of basic research. And I think it's very important. It's not only semantic, I think it's very important. One has to fund basic research because this is what mankind is about. People want to know where they are, where they go, what is their own environment, and they are curious. And this curiosity is leading to research and discoveries that can change the world. So I think that coming back to the budget issue, the only fact that some people in charge of a country have been thinking that it would be a good idea to cut basic research is extremely scary, because it kind of telling us that these people are ignoring history of science and history of discoveries worldwide. So it's not only impacting the US, it's impacting worldwide the mindset that one should have regarding science and funding of science. So that's why I think it's a good time, based on this US story, to really revisit the way we are thinking about funding of basic research, because we need discoveries. There are many problems to take care of. So here at the IUIS meeting, we are talking about health in particular, obviously, but there are other problems for mankind to be solved. And I think innovation is part of the path to solve these issues. So one has to fund basic research. And by the end of the day, that's probably all I have to say, because there's so many examples that is going to be a waste of time. So if some of the people involved in the funding of research can listen to us, I think it would be very helpful for mankind.
So I'd love to understand more of what's going on in Europe. So obviously, U.S. had a change in administration. Typically, when the party currently in power in the U.S. comes in, the president always requests budget cuts. Sometimes it's small, sometimes it's big, sometimes it's more NIH, sometimes it's less. Historically, the worst you get is no funding increase, so you have some inflation erode over time, but Congress, who controls the purchasing, tends to like the NIH funding, often for selfish reasons. It funds a lot of research in the universities in their states and a lot of jobs there. So it tends to be popular, and what the president proposes doesn't happen. Which is fine. And sometimes another government comes in, then they propose huge increases, and that doesn't happen as much either because they don't have enough votes. But, it's interesting because I view the US and the EU as a whole as relatively economically on parity between, if you combine everyone. But it interests me that I always would have assumed naively that the total EU research budget, putting aside military crazy, weird missile things, would be bigger or at least as big as the US, because the US spends a lot of money on crazy missile things. But even in talking and researching for this article, that doesn't, for this talk today, that doesn't seem to be as true. And so I'm curious in learning where the Europe is maybe yesterday before all of the turmoil that's recently happened, what's shifting now in the attitude and how much is changing relative to your point of funding basic science?
Well, I hope it's going to change. As you mentioned, there have been announcements for increasing the budget of research in Europe. Hopefully, it's going to be effective. We have no guarantee of that. But what has been a very important point for Europe research was the European Research Council. ERC really was a game changer in Europe. So this is an institution, an agency of Europe Union, which is funding research. It can be basic research, it can be any kind of research. And, this is the best thing that can happen to a European researcher. I myself was lucky enough to get 4 ERC grants, and it's completely changed the fate of my laboratory, and also the freedom that we could have to explore new avenues, new ideas. Some of them actually have been successful, but not all of them. But it was important to explore them because this is what is our mission. Our mission, I would like to remind that to everybody, is to discover something that nobody has discovered since the inception of mankind. So the bar is pretty high. This is what basic research is about. So in terms of the figures for the budget, I hope for the best. But in terms of the institutions that have been in place in Europe, I think ERC was extremely important. Now there is another agency which is called EIC, which is for the industrial research. So I think that both of these agencies are helping European researchers. Now for France, I think that unfortunately, I don't see any true signs of putting science as a priority in terms of budget. And I think it's really much disappointing considering that announcements have been done at different levels that France, as for Europe, should be a country driven by innovation, knowledge, and research. And for the time being, it's mostly words. And so let's hope that what happened in the United States allows everybody to rethink and revisit what should be the budget of research.
So culturally, why, to me, this is the brain disconnect. The Pasteur Institute, France, right? Like super famous scientist. France has been at the forefront of the Enlightenment and everything else. What changed to make science not just a cultural priority always? Just like missiles in the U.S. are always a priority for our historical quirks, and I can't get rid of that. I'm just surprised that science isn't always there as the foreigner here, so to speak. Just going off of what I think the rest of the world is like based on some cartoon in my head.
Yeah, well, I'm surprised too. I hope I can have an answer to that, because if I had an answer, maybe we could find a solution for that. I think that there is a discrepancy between the words and what's going on, the laws in particular, and the budget. And I agree with you. It's extremely surprising. I think the Pasteur Institute is a very good example of that. France has been at the forefront of research for decades. And despite this leadership role, the effort in research by the government was not at the level of the expectancy. And I'm just going to give you one example, which is Pasteur himself, because Pasteur has to go through what we could call now crowdfunding to actually build the Pasteur Institute. Really. And
the original building, you mean?
Yeah, the original
Paris.
building, which is Rue du Docteur Roux in Paris, 15 arrondissement. And he had to actually beg for money because he didn't have enough money from the government to build it. So I have no answer.
Yeah.
It's just very difficult to understand.
Yeah. I guess it's the fruits of science are not always so obvious, and people just don't see it. It takes a special mind, or it takes some special preparation to kind of put together the dots and say, well, this is this funding that comes today is this benefit that comes tomorrow, or as you also kind of alluded to, there is value, there's intrinsic value in doing this, but yeah, sometimes. Yeah, as a scientist, of course, there's intrinsic value in financing my work and that of my friends around me. So it's a tough sell sometimes, right? Especially when you have so many other things that are also required. A government also has to respond to many other things. Energy. Well, I guess in France it's not so much an issue. The energy prices in other countries in Europe, people are like, why are you spending so much money in funding this research when I have to spend so much money to keep my house warm? So it's a tough sell sometimes, and I think that's so important for scientists to reach out and just transmit this urgency to quote unquote the general public.
No, I can't agree more. I think that there is responsibility from the scientists, from us, of not reaching out to the people and to the public and let them know how important basic research, and to recapitulate on or to tell the story of discoveries and how discoveries have changed the world in health and biology, but in any kind of sector. It's true that when you take, for example, physics, it's very important to say very obvious stuff that lasers were not invented to improve the candles. Okay? Yeah. Lasers were invented because people were curious to understand what light is. And how one can manipulate light. And another example can be taken from Einstein laws by the turn of the 20th century. It totally transformed physics, and one of the applications is GPS.
Yeah.
But he couldn't invent GPS in 1908 or something because he didn't know. So I think that there are many examples of discoveries that are changing our day-to-day life. And I think that, again, to your point, it's the responsibility of scientists to speak about that and to reach out to the public.
I had a weird idea as we were talking. I wonder if America's weird backwards capitalism, which has all its problems, may be helping with the politicians in this case. So what I'm getting at is the politicians get that the NIH funds their universities and then funds research that pharma goes and does, because pharma in the US lobbies like crazy to explain to all the senators to not cut the NIH funding. Otherwise, American capitalism goes kaput. And so while I think, you need the public message, and I think we've been lacking on that, and it's been lacking in the US too, as you can see from politics, there is that other avenue that then leads to these votes that prevent the NIH from getting its funding cut, because the big bad companies, that we all call junk. I work for pharma, so I'm knocking on myself here, but the companies then lobby to the politicians in a way that may not happen in Europe as much, because it's a different, political economic structure. And that's a back door to at least keeping it going. It's a subsidy, right? Because in the US, basic research is considered so risky by industry, they want it publicly funded.
Yes.
And then they take it and go and make money off of it. For better or worse, it does then mean that all the public companies have a vested interest politically through lobbying in maintaining the NIH.
Yeah, but that is, they are not, they have a kind of positive vested interest now in this situation. But then when it comes to, overthrowing EPA rules, they have a vested interest on the other way. Yep. So,
but that's not research. I'm just talking about just the research,
just
the funding
research.
budget to NSF and NIH and all those.
Yeah, but a system that requires that depends on the goodwill of private companies, I think that's also that's also dangerous.
I didn't say it was good. I just said I think that
that's
may
how
be.
It works.
I think that may be the quirk, right?
Yeah.
Because the public in both, in the EU and the US, both are skeptical more of science, and we have a lot of work to do there. But there's this backstop of capitalist interest that's preserving it in the US.
No, it's an interesting idea. One of the reasons why it's not happening, at least in France, is because there are no such examples of big pharmas. There's only one, which is Sanofi. Yeah. So the lobbying is probably much lower than what happens in the United States. And one can also discuss about Sanofi being really French. Okay, that's another issue. But true. This is interesting thought. Yes, maybe.
So. Come on, we were discussing this conversation before. There's one topic that maybe we want to just briefly touch upon, and then maybe we can move more about what is the role of young people in science. But before we move on, so regardless of what the current status of NIH funding is, so it looks like it's not as bad as it initially looked, but a lot of people are starting to feel like this government-sponsored, what would be hardship that's the animosity towards certain research topics might also be problematic. And there's a bunch already of initiatives from European institutions trying to kind of lure researchers from the US to Europe. You mentioned the ERC, so the ERC. So part of the ERC funding, you have the starting grants, the synergy grants that they can provide a couple million for a few years. And those have become, more and more attractive for top scientists trying to establish their own research labs or migrating them. They also get money for relocation that has been increased for people kind of quote unquote escaping America. So I think there's two things here when it comes to topics that have been increasingly harder to do in the United States, and that Europe is trying to kind of position itself strategically. And what should we do? So how much should we or should Europe or European countries be investing in recruiting this talent, and who are going to be these people that come to Europe, in your opinion?
Yeah, but to my opinion, I think that postdocs, not originally from the US, so from Europe in particular, who have been successful in their postdoc training period in the States, actually very good candidates to come back to Europe with the actual climate, so to speak, on the funding of research. Maybe some of these postdocs could have thought of staying in the United States, but considering what's going on right now, I think that several of them, I know some of them, are actually considering not staying in the States at all, but considering other countries, in particular Europe, to come back. And this is where I think Europe should open the door. You are mentioning that some topics are kind of difficult, to say the least, to be studied in the United States. I think that there is no such a fear in Europe for the time being, so that's good. In terms of funding now, across the different areas of research, I think that Europe can provide some kind of funding. You are mentioning ERC, but I know some of the French universities, in particular Aix-Marseille University, has been leading this kind of offers to attract researchers from the United States to come back to Europe or to come to Europe. So I think that this is basically the category of people, of US scientists, who are probably the most likely to be attracted by this position.
Do you think there's a change in attitude as well, kind of a more open arms even to that faculty who is from the US, but maybe like, say, studies environmental science, which you can't research anymore? I'll just say it because I'm the American, I can call it out. And so then there's a drive to go there, because I think the impression from scientists, the old impression of scientists in the US or just people in the US is that other countries don't want Americans because we're, we're a little rude.
No, I can't agree with that at all. And I have example which are speaking for the contrary. No, I think that Europe is very open to any kind of researchers, whatever their origin is and whatever the research program they want to work. And this is one of the strengths of Europe. Now, as we said, the budget of research in Europe is unfortunately not at the level that we expect. So the pie would have to be divided in more parts. But the Aix-Marseille University, they have been offering positions to scientists coming from the United States. So I think this is a good example of the open arm, as you say, from Europe to these scientists. So.
Yeah. I mean, in the end, I think anybody who wants to do science should be given a spot, even if they're American.
Yeah.
We like the American scientists.
Yeah,
They're just used to such big budgets from the NIH that it's like, well, we this is what we got. We do what we can. All right. And so I think we're kind of reaching the end of our conversation, but I think let's just move to one more thing, which is the pipeline of people that come into science, and how are we supporting early career researchers, and what would you, maybe Eric, would like to see in terms of bringing more people to do this important work?
Yeah, I think it's very key. I think that we have to communicate more about what our work is about. Because our mission, as I said earlier, is to make discoveries. And I think that the way science is actually taught at different levels, from primary school to higher school to universities, has to be maybe revisited, because we just don't know what works in terms of making discoveries, but we know what doesn't work. And I think that what doesn't work are walls between disciplines, walls between private sector and academia, and as examples. So I think that one has to educate people on the history of discoveries, to let them know, as we said, that very basic research can lead to very important change in mankind, and also that working together is very important. I think that, as it's said in the United States, united we stand, right? So it works not only for US dollars, but it also works for basic science and for science in general. So I think that one has to establish bridges between disciplines, bridges between academia and private sector that can contribute partially to the funding of research, by the way. This will contribute to attract, or at least to educate people and young people and students to what research is about. And I think that this is, to conclude, also very important to let them know about the beauty of the transformation of basic research into application. I think the health sector and immunology is a very good example of that. And as we started, we might finish by the same example: monoclonal antibodies. Monoclonal antibodies are, have been transforming totally the control of cancer. Now they are used in autoimmune diseases, as we've heard at this conference, and there are many applications to them. And I think this is important that young students are aware of that.
Yeah, for sure.
All right. Well, with that, we're going to do one last gear switch. We're a little bit of a manual transmission today. All right. And so we always end with something that's not quite science related. So the question for you is, if you weren't a scientist, or in your second life, or after retirement, what would you be instead?
Well, that's an easy one, actually. I would like to be a musician, a real one. I am really a very poor amateur, but I'm doing my best. I would like to be an artist, really. There is a famous song in French, which is exactly that. J'aurais voulu être un artiste. I would have liked to be an artist, but I'm not.
What do you play?
I'm playing the bass.
The bass. I play the guitar.
Ah, cool. Good for you.
Also,
I wish I could, but I'm left-handed, and for some reason, I'm playing the guitar and the bass as a right-handed person.
I play as a right-hander, my guitar as a right-hander.
And you're
I'm left-handed, yeah.
Yes. Well, good for you, because I cannot use the mediator. I cannot just do it. You do by fingers only? Okay. Well, I'm not gifted. That's my problem.
I'm sure that you'll be gifted enough to give us a show on Thursday, right? You are part of the band playing at the party. Very much looking forward to that. So who else is playing with you?
Jim Allison.
Luke
O'Neill. Matteo
Iannacone, nice.
And we have 2 singers as well.
Very good.
But I'm sorry, because I'm not sure I will spell their name correctly. So we have 2 singers.
We have 2 singers. That's great. I mean, that's going to be really fun. I'm looking forward to enjoying that in the party.
Be indulgent.
Yeah. I mean, we're going to have 2 free drinks. So don't worry. It's all going to be great.
Just go after the first drink.
Yeah, exactly. Wait, give us time.
All right.
All right. So with that being said, thank you so much for joining us. So we had Professor Eric Vivier from the Aix-Marseille University talking to us about why should we have science and what is the importance of, particularly I think, immunology research, and why in part we are all here in Vienna today.
Okay. Merci beaucoup. Thank you very much.
De rien.
Bye.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at @immunopodcast, Blue Sky at immunologypodcast.com, or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.